Before the cell can enter into mitotic division, interphase must take place. The cell spends 90% of the time in interphase. Interphase is divided into 3 stages. G1. The G1 cycle in interphase produces all materials necessary to replicate chromosomes. S. Using materials created in the G1 stage of interphase, DNA synthesis takes place in the S phase of interphase. In the G2 phase, all materials necessary for the cell to divide are produced. This includes 2 centrosomes, which are important in later mitotic stages. Mitosis is important because it is the process by which growth and development take place. Through mitotic division, 2 daughter cells identical to a parent cell are created. These daughter cells will have diploid number of chromosomes. In prophase, chromatids in the nucleus become coiled into chromosomes. Chromosomes contain 2 sister chromatids. A mitotic spindle made of microtubules begins to form, and centrosomes move towards poles. In prometaphase, the nucleus disassembles. Each sister chromatid develops a kinetochore that attaches to the spindle fiber. In a later stage, the 2 sister chromatids are separated from one another via the spindle fiber, centrosomes, and kinetochores. In metaphase, centrosomes reach the 2 poles. Chromosomes gather at the metaphase plate in the middle of the cell. Kinetochores attach to each sister chromatid and become connected to the pole via the spindle fiber. Anaphase is the shortest stage of mitosis. Sister chromatids are ripped apart and move toward opposite poles via spindle fibers. During this stage, the cell elongates to allow for future division of cytoplasm. In telophase, chromosomes become uncondensed. At the metaphase plate, the cell begins to cleave and cytoplasm is divided between the daughter cells. A new nuclear envelope arises in each daughter cell. We now have 2 genetically identical cells. Meiosis is important for the creation of sex cells, which join together via fertilization to create life. Meiosis creates cells that have a haploid number of chromosomes. Meiosis, unlike mitosis, creates 4 daughter cells that are not genetically identical to the parent cell through 2 separate stages of division. Similar to mitosis, meiosis first goes through interphase, where the cell spends the majority of the time replicating genetic information and preparing to divide. Meiosis 1 then begins. In meiosis 1, homologous chromosomes become separated. Genetic variation also takes place in meiosis 1 via crossing over.
In prophase 1, crossing over takes place where genetic information from 2 non-sister chromatids switch chromosomes. This creates genetic diversity. Synapsis also occurs where homologous chromosomes condense and are paired together. Centrosomes also begin to move toward poles. In prometaphase 1, the nuclear envelope dissolves. Each chromosome in the homologous pair develops 1 kinetochore and spindle fibers begin to develop. In metaphase 1, homologs independently assort as they line up on the metaphase plate, connected via their kinetochores in the spindle apparatus to the centrosomes, which have reached the poles. In anaphase 1, homologs are pulled toward one pole or the other via microtubule-constructed spindle apparatus. This created 2 haploid cells from 1 parent diploid cell. In telophase 1, division of the cytoplasm occurs. Interphase and reconstruction of the nuclear envelope rarely occur before the 2 new cells move into meiosis 2. 2 haploid daughter cells are produced. In meiosis 2, sister chromatids are separated from one another by mechanisms similar to those in mitosis. In prophase 2, chromosome number is now haploid. Each chromosome contains 2 sister chromatids. A mitotic spindle begins to form and centrosomes move towards their own poles. In prometaphase 2, each sister chromatid develops a kinetochore. In a later stage, the 2 sister chromatids are separated via the spindle fiber, centrosomes, and kinetochores. In metaphase 2, centrosomes reach the 2 poles of the cell. Chromosomes gather at the metaphase plate to be pulled apart by spindle fibers and kinetochores, which are now connected to one pole. In anaphase 2, sister chromatids are ripped apart and moved toward opposite poles via spindle fibers and protein motor movement. In telophase 2, cytoplasm is divided to share by daughter cells, for creation of a total of 4 haploid genetically different daughter cells.